One of the most common ways that microbes exchange genes is through horizontal gene transfer. This is an amazing process, whereby one microbe produces a free-floating piece of DNA, usually a gene or a set of genes. They're packaged together or somehow transported to another microbe where they can be exchanged. The receiving microbe can integrate that DNA into its own genome and express the proteins. Not only can this happen deliberately with two microbes working together to exchange DNA, but free-floating pieces of DNA can also be picked up from the environment and integrated into the genome. This horizontal gene transfer is an incredible phenomenon, and it has huge ecological implications. For example, if a new pollutant enters a habitat, the local soil or marine microbes can evolve to break down the pollutant. This gene can be shared with other microbial populations and species, and in this way, entire microbial communities can cultivate these genetic abilities on a rapid timescale. You don't have to wait for every individual strain to evolve it on its own. This kind of gene transfer is one of the only known mechanisms by which organisms can exchange DNA in a way that doesn't involve mating or direct ancestry. Now this brings me to today's news. There's a pretty crazy animal called Placabrancus Oscillatus. It's technically not an individual species, it's a species cluster or a species complex, consisting of multiple closely related species of sea slugs. These sea slugs feed on algae, and of course the algae contain a number of genes involved in the production and maintenance of chloroplasts. What's really crazy about this animal, this species cluster, is that these sea slugs have somehow integrated these chloroplasts into their own bodies, that it's a gene transfer of algal genes into the cells of a sea slug, of an animal, of a vertebrate eukaryote. Super weird. And somehow, through a mechanism that isn't understood, these integrated chloroplasts are still able to function. This is really strange, because when these chloroplasts are, naturally grown in the algae's genome contains a plethora of genes that are critical for the maintenance and regulation of these chloroplasts. The proteins used in the chloroplasts also apparently have a pretty high turnover rate, so you would think that removing the algae nucleus would quickly lead to the collapse of the chloroplast. After all, what's producing the proteins to replace the proteins that are being quickly worn out? Well, in the sea slug, when the chloroplasts are integrated into the animal's tissue, the algae cell nuclei, which contains the algae genome, isn't brought along. The algae genome is left behind to be digested in the sea slug's gut, along with the rest of the algae. So somehow, these sea slugs are preferentially selecting and integrating the algae chloroplasts, and they're somehow keeping the chloroplasts intact and actively photosynthesizing for months. A new study has recently been published in the journal eLife, which sought to explore the relationship between algae chloroplasts and their sea slug hosts. They also wanted to analyze the genome of these photosynthetic sea slugs in an attempt to understand just what exactly is going on here. One major part of the research was a starvation test. They reduced the food intake for a number of experimental sea slugs to put them into a starved state, and then, while still deprived of food, exposed them to varying light levels. They found that the slugs could, via these chloroplasts, perform photosynthesis and produce oxygen and sugars for more than 3 months before the chloroplasts finally degraded. Amazingly, the efficiency of the chloroplasts in the sea slug tissue was not significantly different than it was in the original algae. So these are basically animals that are just as photosynthetically efficient as the original algae. Another major part of the research was a genetic analysis of the sea slug, which was kind of tricky because there haven't been too many previous genetic analyses of these animals, so the genetic reference databases in this Isle of the Gastropod section are pretty empty-shelled. Despite this, they were able to make an important discovery. The scientists thought that they would find evidence of horizontal gene transfer, where the sea slug cells have somehow developed a way to steal chunks of the algae's DNA, which lets them express these proteins and construct and maintain chloroplasts on their own. But to their surprise, this is not what they found. In fact, they found no evidence at all of any genetic sequences from the algae having been integrated into the sea slug genome. This means that horizontal gene transfer is not what's happening here. I'll quote Taro Maeda, one of the authors of the paper. He said, quote, "We are embarking upon a new challenge to answer the question, how does the sea slug retain this function without horizontal gene transfer? Our genome data also provides clues to this. We have found several candidate genes related to the long-term maintenance of photosynthetic activity. These genes, related to protein metabolism, oxidative stress tolerance, and innate immunity, should be subsequently highlighted in future studies." Unquote. Okay, this is super fascinating. After integrating the chloroplasts into their cells, the sea slugs don't also utilize algae genes. Instead, they upregulate some of their own genes, involving genes for, quote, carbohydrate metabolism, proteolysis, and immune response, unquote. In less technical terms, this means that the sea slugs are using their own genes for processing the sugars produced by photosynthesis and for breaking down and recycling the proteins used in photosynthesis. The upregulation of immune response genes could have several explanations. This might be some kind of safety mechanism to protect the sea slug from any pathogenic effects of the algae chloroplasts, or it could be that some of the immune response genes have secondary or alternative functions useful for maintaining the chloroplasts, or it could be a combination of these or other factors. The really big, incredible, mind-blowing takeaway here is that scientists have discovered a mechanism by which a complex adaptive trait like photosynthesis can be transferred from one organism to another without the use of traditional horizontal gene transfer. While this is awesome, it's certainly a rare event, and it clearly requires a complex toolkit of genes that are at least somewhat compatible with the new system being installed. There's still a lot that needs to be learned. A lot of the granular mechanisms and molecular interactions in this process are not understood. They've ruled out horizontal gene transfer, but that really only deepens the mystery. At the chemical level, how is this happening? What are these upregulated sea slug genes doing precisely to sustain the chloroplasts for months at a time? All valid questions that can lead to important discoveries. For example, if we figure out how this process works, we might be able to artificially install chloroplasts in other animals and essentially create genetically modified photosynthetic animals, like cattle that can make sugar when exposed to sunlight, and so might require less feedstock, or birds that can make sugars while in flight and can thus enjoy less selective pressure and competition for food. This is super fascinating stuff, with super fascinating, almost, borderline science fiction implications.